Okay, so today we're going to be doing proteins. Proteins serve many different purposes in the body, and they come in many different types, such as enzymes, which catalyze reactions in the body, structural proteins, and transport proteins.
All right, so in proteins there are amino acids, and there's 20 amino acids overall, and they all have one thing in common, in which they have carboxyl and amino groups. And they also have an R group, which determines different properties such as hydrophobicity. And here are serine and cysteine, and both of those are polar, and they hydrophilic amino acids. So they interact with water, and with cysteine, it has the R group of sulfur and bonded to a hydrogen, which makes it very reactive. And serine has an R group of hydroxyl.
Okay, so I'm going to be describing 4 different amino acids. One of which is aspartic acid, which is the amino acid with a acidic R group. Alanine and glycine are the 2 hydrophobic amino acids which don't interact with water and are nonpolar. Alanine in specific, its R group is methyl, and that is the group that's making it nonpolar. And for glycine, the R group is hydrogen. And then we have, for the aspartic acid, the R group is a carboxyl group. And then there's lysine, which is the basic amino acid with a basic R group. And the aspartic acid and the lysine, they're acidic and basic, and because of that, they're both charged polar.
Okay, so amino acids are the monomers which form polymers called peptides, and they're formed by a series of dehydration reactions. And in amino acids, the dehydration reaction occurs between the carboxyl group of one amino acid and the amino group of the other. So just to demonstrate here, I'm taking the hydroxyl of the carboxyl group in one amino acid and in a hydrogen from the amino, and that forms water. And then we bind the carbon to the nitrogen, and that is the dehydration reaction. So in amino acids, it occurs specifically between a carboxyl and an amino, which is called a peptide bond.
So this ties into the primary structure. So here is a polypeptide chain, which is a string of amino acids, and these strings of amino acids are often 100 to over 1,000 amino acids in length. And it's a specific sequence of amino acids. And additionally, the R group determines the formation of the secondary structure, which is important to know for later. And in the model, each different colored gumdrop represents a different amino acid.
Okay, so the next structure in terms of the hierarchy of proteins is secondary. And so secondary structures, basically, they have, you can see the local folds within them through the hydrogen bonds. And so they're found either as alpha helixes or beta pleated sheets. And so you can see you have an alpha helix here, which you can see the hydrogen bonds represented on toothpicks with the 3 dots. And then you also have, in this form, where this should be more wavy and foldy, but this is also how it forms. And so then the next step in the hierarchy would be a tertiary, which is basically a long chain of both beta pleated sheets and alpha helixes combined together. So here you have an example of a tertiary structure. And so then if you, for then quaternary, which is next, you would take another tertiary, you would combine these 2 and then add it to that, and that would be an example of a quaternary. And also, another way of looking at it is something similar to this, where you can see that the different colors on the belt represent different long chains that are bonded together in terms of quaternary. Quaternary is basically just a long amount of tertiary protein structures.
Just one thing to add is that proteins in their tertiary structure can either be globular or fibrous. In globular proteins, they basically just have a bunch of alpha helices and beta pleated sheets kind of wrapped around each other. So this would extend and kind of go in circles around that. And there's also fibrous proteins, which are more organized and evenly spaced out. So now I will be describing the denaturation process which is basically when a protein loses its shape, and this can be caused by excessive heat. So here we have an alpha helix, and in denaturation it would unravel like so, losing all of the folds, and this would continue to basically unravel the whole thing. And now the protein has lost its structure, which means it has also lost its function, as structure implies function.